All right, we are recording. I'm going to get off of this screen and go to the PowerPoint now. Can someone confirm that what you're looking at is the PowerPoint slideshow, please? Anybody? Bueller? That's an old reference. So, no, hopefully this is working. Can someone confirm you can see the PowerPoint slide?
I can see it.
Perfect. Thank you. All right. So again, it's a little bit early, so I'll just go ahead with the redundant announcements, the nothing new ones. I don't mind starting early for that. So as always, I remind everybody to check your emails and course announcements before you come in to make sure that we're actually meeting in person. Today would have been a good day for that, even even though we had planned to meet online, but my daughter had missed the bus and I was behind, so that's the kind of, and we have snow, obviously the first snow of the semester. So that's the kind of stuff you got to look for. Make sure again that we're meeting in person before you come in. Also, I'll remind you, my office hours today are online only. I'll be online at least from 9 to 10. Usually you can find me a lot, much hours past that, but probably not today. I will be celebrating the Marine Corps birthday. So, yeah, any questions about office hours? All right. Speaking of office hours, if you meet me during office hours or outside of office hours, if you need to get feedback for exam 1 and exam 2, you can get all the correct answers, which you're going to need to help study for the final, and which is going to be on a Monday, early December at 8 a.m. Anyway, you'll get a 5% boost in addition to the correct answers. Any questions about that? All right. Exam 3 was supposed to close yesterday, but one of your classmates noticed there was a glitch. And for some reason, it closed early, like 7 something in the morning, I think, yesterday. But they let me know pretty quickly, and I fixed it right away. So you shouldn't have been affected. But just in case you were affected, in case you're one of those kind of students who went and saw that it was closed and said, "Oh, well, there's nothing I can do," and left it at that. Well, I've reopened it. It's open until today at 10:30. So if you haven't taken the exam yet, after the lecture, you'll have time to take it. So any questions about exam 3? All right, and once it closes, you'll be able to retake it as usual. But anyway, I also want to remind you that this week's lab is virtual. All you have to do is watch a video, a documentary. So we're not going to meet in person. Next week's lab will be virtual, but we are meeting in person. We're going to do it together, sort of like we did last week. Of course, if you get it done before that time, then you don't need to come in. But anyway, are there any questions about labs for this week or next week? All right. As always, attendance is mandatory. Participation is graded by 9 a.m. If you're online with me right now, you're going to need to send an email that has all the attendance words or phrases, and then also one question about the material covered. All of that to get full credit. Actually, extra credit. It's one of those things, even though it's required, you all still are getting extra credit for doing it. Or if you're only getting partial, some of the words or questions, and partial credit. So any questions about announcements or reminders? Does anybody have your own announcements or reminders that you want to share with us? All right. Well, before we get started, then the first word for attendance is going to be, well, letters actually, USMC, United States Marine Corps. That's what it stands for. And again, it's today's the Marine Corps birthday. So that's the first word for attendance. Let's get moving. So we left off talking about, we're kind of looking at evolution from a wider perspective, so not just natural selection, right? We're looking at the other ways that evolution occurs. And the last thing we were talking about was sources of genetic variation, and we talked about mutations. And you're already familiar with mutations, because you learned about that in a previous chapter. But yeah, that was the ultimate source, and that's the important thing that you needed to know for the exam, right? Mutations are the ultimate source of genetic variation. But that leads us to another source of genetic variation, and you wouldn't have this other source without mutations, but nonetheless, the next one is sexual reproduction. So for sexually reproducing organisms, most genetic variation in the population is from a unique combination of alleles of each individual inherits. Actually, you know what? Now that I think about this, let me double check. Okay, yeah. So there's a fresh assortment of existing alleles that arise from every generation from 3 random components, as you already know. So you have independent orientation of homologous chromosomes, or in other words, a shorter way of saying that, as you already know from the previous chapter. That's just independent assortment, right? So basically, if you remember, that's saying when you're in meiosis of, and all your chromosomes line up in the middle, all those homologous chromosomes, it's not like it's all mom chromosomes on one side and dad chromosomes on the other, right? It's completely random. And because you have 23 pairs of chromosomes, that means there's millions of different ways that can happen, right? So there you're already looking at millions of different varieties when it comes to sexual reproduction and having this genetic variation. And then that doesn't even include the next one, which was crossing over. So remember, those homologous chromosomes, when they're next to each other, sometimes they will swap chunks, right? So you might have a dad chromosome swapping a chunk with a mom chromosome. So then you're left with a dad chromosome, little mom chunk on it. And then conversely, on the other hand, you'll have that mom chromosome with a little dad chunk on it, right? So it doesn't always happen. Sometimes it happens more than once. Sometimes it's a big chunk. Sometimes it's a small chunk, right? So it's completely random. So again, that's another way we get our genetic variation. And of course, finally, random fertilization, right? So the chances of sperm X getting together with egg Y, for example, right? It's completely random. So anyway, that's all old news. We're just saying it again in the context of evolution, as opposed to in the context of meiosis. So any questions about this slide? All right. So let's talk about this important point: populations as the units of evolution. That will be a test question. Before I even get into this slide, I'll go ahead and tell you there will be a test question where you need to know individuals do not evolve. I know I said that last time we met, and I used the black moths as an example, right? There was no single moth that went from light colored to dark colored, right? So individuals do not evolve. Now, individuals do go through some changes, right? That might happen, but that is not technically evolution. By definition, it's only populations that evolve. So that being said, now let's dive into this and look at it in more detail. But that's the big picture. That's what you need to know for the exam. So again, the evolutionary impact of natural selection is only apparent in the changes in a population over time. As a matter of fact, I think I should update that to say the evolutionary impact of evolution. So not just natural selection. Excuse me, I've got to turn something on here. Okay. Yeah, so it's again evolution, right? And populations are the unit, so to speak, of evolution. So I guess that brings up the next question. And I've mentioned this before, but oops, here it is again. I can get down to it. There we go. What is a population then, right? So if populations are the unit of change for evolution, then what are populations? A population is a group of individuals of the same species that live in the same area and interbreed, or at least potentially interbreed. So a good example for that might be the stray cats that live in your neighborhood, right? That would be a population. Now, that would be a different population than the stray cats that live in my aunt's neighborhood in Key Largo, right? Because that's a completely, there might be the same species, but they're in completely different areas. So they're not interbreeding. They're not competing for the same resources, right? So that even though they're the same species, they're not the same population. And people get confused with this, I think, often because I think outside of biology, when you talk about population, people often think naturally of humans, right? The human population. And basically, for the most part, humans, at least potentially, could go all over the world, right? So humans, in a sense, are potentially interbreeding with the entire human population, right? But that's not like that when we're talking about biology and we're talking about other organisms, right? Other species. So just keep that in mind. That's what a population is. There'll definitely be a test question about that. And then actually in the chapters that come after this, we will bring up the concept of a population again. And again, you're going to need to know the definition. So any questions about what a population is? All right. The next word for attendance, and I want to remind one more reminder, sorry. As usual, so if you're with me right now, you don't need to worry about this, but if you're watching the video at a later date, I just want to remind you that you do need to do a screenshot every time I do one of the attendance words. But anyway, the next word for attendance is going to be Philly, as in short for Philadelphia. I'll just put a P here. So the screenshots to show that weird looking P. Anyway, yeah, Philadelphia. It's the birthplace of the United States Marine Corps. So anyway, Yeah, so different populations of the same species may be geographically isolated from each other. This is what I was getting at earlier, right? Just because they're the same species doesn't necessarily mean they are the same population. And because of that, you get the exchange of genetic material may never happen, or at least very rarely happen. Um, and such isolation is common in populations confined to different lakes or islands. And a good example of that, and I wanted to bring this up because here we have lakes or islands, but island in this context does not necessarily mean a piece of land surrounded by water. Now, granted, that is the definition of an island, and that's usually what you mean, even in this context. But there are exceptions. And one example of that is and this is not going to be in your test book, but here in West Virginia, near the Cranberry Glades area, the mountains are so tall that it gets really cold up there. So the very top of some of these mountains, there's plants that you will only find in Canada, other than at the tops of those mountains. So in a sense, the tops of those mountains are islands, right? Biologically speaking, because those organisms, those plants, are confined to that spot. So they're surrounded by a sea of in uninhabitable habitats. So anyway, that's just an example. Here's some book examples on the next slide. So again, these when we're talking about populations, again, yes, they're the same species, but they also have to be basically living together, right? Interbreeding, dealing with the same issues, competing for the same resources. And this picture is a great example. So imagine, if you will, if it was, I don't know, 500 years ago, and maybe there was more water, right? Maybe the water has receded in 500 years since 500 years ago. I try to imagine this, instead of having all these multiple lakes, imagine there's just one lake, right? And then you just have this one population of a specific species of fish. But then imagine the water starts to recede, and now you end up what we have here, right? All these different lakes. Now, in each of these lakes, you might be able to go in and find whatever species, that hypothetical species I was mentioning earlier. They might all be the same species, but they're different populations, right? So they're dealing with different things. They're not interbreeding. For example, this lake right here, to me anyway, I'm not sure, but it looks really shallow and small. So I would assume this lake has very warm water, and this lake might be deeper and have cooler water, right? So all these different things. So again, same species, different populations. They're dealing with different things in their little isolated habitats. And even though that's not the point of what we're talking about right now, I do want to mention too, or remind you, when we talked about allopatric speciation, right? This is the kind of stuff that might create new species, right? So right now there might be the two different populations of the same species, but eventually, after generations and generations of living in different environments, that might result in a new species. And then one other thing I want to point out too before we move forward is this little bullet point right here. Exchange of genetic material may never or rarely occur. And this is just another great example for that, right? So you can imagine, for the most part, there's not going to be some fish jumping out of the water from one lake and flopping its way over land to the other, right? That's not going to happen. But what might happen is you might get some sort of, I don't know, like an eagle swoops down, grabs a fish from one lake and is flying off with it and accidentally drops it into the other lake, right? That would be a rare exchange of genetic information. Or even a human, right? Maybe humans going fishing in this lake that I circled right here, and then they come over here and they do a little fishing there, and they decide they don't want to keep their fish, so they throw them in that lake. So again, it's not like these populations never mingle. Potentially that might be the case, but there's also potentially the case that they do rarely mingle. So any questions about that concept, this idea of isolated populations? All right, that brings us to another term that you're going to need to know. At the population level, which is what we're talking about here, right? Populations are the unit of evolution. Biologists focus on gene pools, which consist of all copies of every type of allele at every locus in all members of the population. So in other words, it's like, hopefully you're already familiar with this term since we've already passed it, but your genome, for example, right? That's all your genetic information, right? That's all your DNA, all 23 pairs of your chromosomes, also the DNA that comes in your mitochondria that you got from your mom. All that is your genome, right? So if we're talking about a human, that's your genome. But if you expand that to a population, instead of calling it a genome, you're calling it the gene pool, right? So that's everybody's genome. So any questions about what a gene pool is? In case you don't get the picture reference, that's the kid's name is Gene Belcher, and obviously they are at a pool. Anyway, if there's no questions about gene pools, we'll move forward and talk about analyzing gene pools. Now, this is good in a sense. On one hand, I wish we could have talked about this before lab last week, because that's what you did in lab last week. But on the other hand, I guess it's nice that you've already done it in lab. So now this might come a little bit easier for you in lecture. So let's talk about analyzing gene pools. Now, before we do that, I'll remind you too, in the experiments we did last week with the Hardy-Weinberg, we were quantifying changes in the gene pool, right? We were we quantified like what percentage are the dominant allele and what percentage are the recessive allele, right? And we looked at how that changed over time. That's the only calculations we did, right? We didn't calculate, for example, when we started, most of the experiments were 50-50, right? So 50% big R and 50% little r. We didn't calculate that. That was given to us in the simulation. And then we ran the experiment for 100 generations. And then once again, we got the percentage of big R's and the percentage of little r's, but we didn't calculate that. Once again, that was given to us. The only thing we calculated was like, all right, well, we started here, we ended here. So this was the change, the total change, and then this was the percent change. So that is a little bit different. That's like a very basic version of what I'm about to teach you, because what I'm about to teach you isn't necessarily quantifying starting here and ending here necessarily, even though I will talk about that. But it also talks about, well, just the calculation itself of coming up with this. All right, it's again, 25% big R, 75% small r, or I don't know, 25% big R, 50% big R, small r, 25% little r, right? Those are the kind of things that we're going to talk about in this section. But that being said, let's jump into it. So imagine, if you will, you don't even need to, you can look at this picture, a wildflower population that has two varieties of blooms that are different colors. So you don't need to imagine it. There you go, right? There's a wildflower population, there's red flowers, and there's white flowers. So let's assume the allele for the red flower is dominant to the allele for the white flower. So we'll just randomly, arbitrarily call it big R and little r. So the genotype frequencies can be calculated from allele frequencies if the gene pool is stable. And we'll talk about that later, the whole idea of a stable gene pool. So for now, just keep that in the back of your mind, and we'll get back to it. But so again, basically what we could do is use algebra to determine these frequencies, right? And remember, a frequency is another way of saying it's it's another way of saying a percentage. Of course, with the frequencies, when we use it mathematically like that, the only difference is we don't actually use the percent sign, right? And we don't move the decimal place twice to the right. But anyway, another way of looking at a frequency is you could say it's almost like a ratio, right? And what I'm getting at with all that is, when we have these equations, they always need to equal 1. So, and again, I'll use the goldfish experiment from last week. I remember one of them we had, and I just said this verbally, but now let's put it in writing. Remember, we had 25% was big R. We had about 50%. So in this case, since it's a frequency, we're right at 0.5 was big R, little r. And even though we already know the answer, I'm going to go ahead and put it to you like this, because again, we're talking about the Hardy-Weinberg formula. So I'm going to look at this algebraically. So this is the kind of easy question you might get. So I might say something like, again, big R is dominant, little r is recessive. And I might say something like, so there's, 25% of the population is big R, right? I would give you that information on the exam. And then I might say, and 50% of the population is big R. Wow, that's horrible looking. Sorry. Big R, little r. So then you might get the question, all right, what percentage of the population is little r? And again, this is just basic math, right? So it always has to equal 1. So you got 25 and 50. That makes 75, right? So that means this must be 25. So that's the kind of basic questions you might get. But yeah, that's the Hardy-Weinberg formula. If you download the PowerPoint, you can click this link and try to remind myself what it is. Yep, it's a little YouTube video that explains it, explains how we got it and how to use it. But yeah, that's how that works. So again, we basically use algebra to figure out the percentage, the frequencies of homozygous dominant, homozygous recessive, heterozygous. And let's look a little bit more at it. So there it is, right? That's the equation I just used. And of course, the even easier version, right? Everything I've been talking about is the genotype frequencies, right? So again, homozygous dominant, heterozygous, homozygous recessive. That's the genotype frequencies. That's a little, slightly more complicated. But the really easy one is the allele frequencies. And again, it just has to equal 1. So you might get some kind of test question if I say, I don't know, 80% of the population, or yeah, of 80, and good grief, in this population, 80% is big R. And then I might say, so what percent is little r, right? And hopefully you don't even need a calculator for that. I hope it's nice and easy, because what plus 0.8 equals 1? And the answer is 0.2. So this is the kind of easy math questions you might get on the exam. So any questions about that? All right, another question you're going to get on the exam that doesn't require math, but still is about this this equation, is it might say something like, in the Hardy-Weinberg formula, what does p squared represent? And that's dominant, right? Homozygous dominant. You need to know that. What does 2pq represent? And you need to memorize that represents heterozygotes. Or I might ask you finally, what does q squared represent? And that represents recessive homozygotes. Now you might be wondering too, like, what's up with the squared? Why all that? If you're not very familiar with algebra, you probably are thinking that. And when I did that really simple formula as an example previously, I didn't do anything with squares, right? So a little bit later, and then in the following slides, I'm going to show you a more complicated way of using this algebraically. But I'll go ahead and tell you now, that complicated version where you need to take square roots and things like that, we're not going to be doing that on the exam, right? So you should not need a calculator for the exam, because it should be very basic math, like the examples I've given you. But are there any questions so far? All right. The next word for attendance, it's a name. It's Samuel, or just make it Sam for short. For Captain Samuel Nichols, the first Marine Corps recruiter way back in 1775. Anyway, if there's no questions, let's move forward. Yeah, so like I said, that was the basic version. Next, I'm going to give you a little bit more of a complicated example. But again, this is just an example. We're really going to be digging in a little bit more with algebra with this example. Again, taking square roots and things like that. But for this 100 level non-biology major course, I'm not going to require any kind of algebra at that level. But let's talk about how it's used. So we can use the formula to figure out stuff about health science. And an example that the old textbook gave is this disease called PKU. By the way, this is just an example. I'm not going to ask you any questions about PKU, but really quickly, just so we can talk about it, it is the inability to break down a certain amino acid, right? So if you have this disease, you cannot break down that amino acid, which is not good, right? It's caused by recessive allele, right? So that means, like I taught you earlier, we're talking about these Q squared here, right? That's the people who have it would be homozygous recessive, right? Because again, it's a recessive allele, and if it's a recessive allele, you have to have 2 copies to have the disorder. So that part that is relevant in this example. The other thing that's relevant in this example is that there are 1 out of 10,000 babies in the U.S. are born with this, right? So again, putting all this together, we know that 1 out of every 10,000 babies in the U.S. is homozygous recessive. That's all we know. So with that, we can calculate other things. And here's how we'll do it. So again, anybody who has that has to be homozygous recessive. So they are identified by Q squared, right? And when you write 1 out of 10,000, you get this number, right? So 1 divided by 10,000 gives you this. So that's what this means, right? Q squared equals this. That means, again, this is the percentage, so to speak, of humans in the U.S. who are born with this recessive disorder. So now that we know that number, then we can use some algebra and figure out the other number. So what we'll do is, first of all, we need to figure out what Q is. So if Q squared is this, then we take, even though you can't see it, basically we're taking a square root of both sides, right? So the square root of Q squared is Q. The square root of 0.001 is 0.01. So now we know what Q is, right? Q, if you could write it here, that might get a little bit sloppy, but it is 0.01. Whoops. Anyway, so now we know what Q is. And then remember, even though it's not on the slide, the other equation was even simpler than this, right? It was just P plus Q equals 1, right? So we just figured out what Q is. So then we can just algebraically figure out what P is. So obviously, again, this is basic algebra. P plus Q equals 1. Therefore, subtract Q from both sides. P equals 1 minus Q. Hopefully you get that algebraically. And another way of writing that is 1 minus Q equals P, right? Which is what you have at this bullet point right here. So then, once again, you just do the math and you can figure out that P is 0.99. So now we figured out what P is because we figured out what Q is. And then once you know what P and Q are, then you can plug that in to figure out how many people are heterozygotes. So all that, we now know that about 2% of the population are carriers. And we figured that out just by knowing the Hardy-Weinberg formula and knowing that 1 in 10,000 babies are born with this recessive disorder. So are there any questions about this slightly complicated example that you will not need to do on the exam? All right, nice and easy. So we'll move forward. Now we're going to talk about microevolution as that causes the changes in the gene pool. So once again, this is something that you're already familiar with from the lab, right? We already did this. But there will be some new terms. So for example, a non-evolving population is known to be in something called genetic equilibrium, aka Hardy-Weinberg equilibrium. And I'll go ahead and tell you now, this is all hypothetical and academic. So because for the most part, as you're going to see when we talk about it a little bit later, for the most part, this is not even a real thing. For the most part, populations are going to change, right? From generation to generation, things are going to change. But again, focusing on this hypothetical idea here of the Hardy-Weinberg equilibrium, that would mean that the gene pool remains constant from generation to generation, right? So again, using those goldfish as an example, that would mean, if there was 25% of the fish were orange with white spots, then generation after generation, that percentage would remain. There would be no increase or decrease in that percentage of spotted fish. And another way of saying that bullet point up here, since we're talking about Hardy-Weinberg, to say that mathematically, you would say this is the same thing. We're just saying it mathematically, right? The frequencies of alleles, P and Q, and genotypes, P squared, 2PQ, and Q squared, remain unchanged, right? So those two things are saying the same thing, just in a different way. And because generation to generation change in allele frequencies is evolution on the smallest scale, this is why we refer to it as microevolution, right? So again, that experiment that we did, those experiments that we did last week, that was microevolution. Those fish weren't changing, right? The population, that species didn't change to a new species. Even the types of fish didn't change, right? There was only 3 types of fish: orange, white, and spotted. Now, the ratio of how many orange to how many spotted to how many white, that changed, but the fish themselves, right, they didn't change. So this is, again, microevolution. I just want to reiterate that because, once again, like I said last week, a lot of people who oppose natural selection or evolution, that's usually why, right? Because what they're actually usually opposing is the idea of new species arriving. So again, this is not new species. This is just the same old species, just something within the species changing. So are there any questions about this slide before we move forward and talk a little bit more about the same concepts? Okay. So let's talk about these different mechanisms of evolution. So again, the big one is natural selection, and we've talked about it a lot. And we're going to talk about it again. But then there's these other ones that we did deal with in the lab, right? So we did deal with genetic drift. We did deal with gene flow. We did deal with mutations, and we did deal with natural selection. So here we go. Let's jump into it. So, what mechanisms can change a gene pool? Again, like I said, natural selection is the most important, but there are other ones. So, let's talk about this one first, genetic drift. For the exam, you need to know what genetic drift is. I might ask you the definition, or I might give you an example of a scenario, and you need to recognize that as genetic drift. But yeah, genetic drift is a change in the gene pool due to chance. I should put pool there, right? It's not a change in gene, a change in the gene pool due to chance. And again, using the example from last week's lab, remember we set up the parameters for that first experiment to where there was no advantage to being white or orange or spotted. There was no disadvantage to being white or orange or spotted. There were, right? So we ran the simulation and the population did change over time, but it was completely by chance, right? Completely by chance. There was no selective pressure to make one color more prevalent than the other. So here's an example from the old textbook. Here we have another wildflower population, white flowers and red flowers. And again, this is genetic drift, right? So there is no advantage to being red. There's no disadvantage to being red. There's no advantage to being white. There's no disadvantage to being white. These are flowers too. So there's no sexual selection necessarily. I guess that was a bad example. I should have said it like that. But anyway, let's just assume there's also no sexual selection here either. So what happened is you can see this original population on the left. You can even see the numbers, right? So 70% of them, 70% of the gene pool is big R, right? Therefore, 30% is small r. Now, in this generation, the ones that are circled here, those are the ones that just happen to mate. And again, it's completely by chance. There's no advantage or disadvantage here. So by chance, those are the ones that happen to mate. Therefore, this is the next generation. By chance, the next generation, generation 2, is 50-50, right? When you look at their gene pool, 50% of the alleles are big R and 50% of the alleles are small r. And again, that was completely by chance. There was no advantage or disadvantage to making this happen. Now, again, in this generation, they reproduce, and it just so happens to be this one reproducing with this one. Again, by complete chance, there's no advantage or disadvantage. So when those mate, obviously, I could ask you this question, but I don't want to waste the time waiting for somebody to answer. So I'm going to move forward. But if I were to ask you, what would be the result of this mating? There's only one option, right? They would all be big R, big R. So there we go. After 3 generations, by complete chance, we are left with nothing but red flowers, right? All the white ones have gone extinct. They're gone. And again, that was completely by chance. That's genetic drift. Does anybody have any questions so far about what genetic drift is? All right. And to remind you, I know I'm being repetitive here, but again, this is just an example. So when you're studying, don't feel like you need to memorize this. Like there won't be any test question where I say, hey, remember that slide with the white flowers and the red flowers? What was the population? What was the percentage in generation 2? Nothing like that. Now, granted, I might give you this picture on the exam and ask you to interpret it. So yes, I might give you this picture and say, hey, well, what percentage of the gene pool was big R in the second generation? And you can just look and say, all right, well, there's the second generation. He wants to know the for big R, and it's 0.5, right? Nice and easy. So any questions? Okay. Let's talk about the bottleneck effect. And before I do, I want to point this out too. For the exam, like I said, you're going to need to know what genetic drift is. You're also going to need to know the difference between the 2 subcategories, right? So there's 2 different within genetic drift, there's something called the bottleneck effect and the founder effect, which obviously we're about to discuss. And you will need to know, excuse me, that both of those represent a different type of genetic drift. And you'll need to know, the differences between the 2, like how are they different and how are they similar? So let's talk about it. And hopefully it's going to be an easy concept for you because it's kind of in the name, and that'll make sense when I show you a visual. But again, the bottleneck effect is another example of genetic drift. And the thing you need to know about it is that second bullet point right here. It results from a drastic reduction in population size. So obviously, even though the textbook doesn't get into it much, and neither does this PowerPoint, the drastic the drastic reduction in population size would obviously need to be by something random, because again, we're talking about genetic drift. So it can't be, for example, if there was, I don't know, a new disease came into a population, and it killed off most of the population, the weak ones, but the ones that are left, that had the genes for it, that would be different, right? That would be a drastic reduction in population size, but that would be natural selection, right? Because nature eliminated all the individuals with the alleles that made them susceptible to that disease. So keep that in mind when we're talking about a bottleneck effect. Yes, it is a drastic reduction in population size, but it has to be random. But anyway, and that's why it gets its name, the bottleneck effect, right? Because the population, in a sense, is passing through a bottleneck. And again, I think that's going to make a little bit more sense when I show you the visualization in an upcoming slide. So what happens here when a population goes through a bottleneck, and again, you've lost most of the individuals in the population, what happens then is you decrease the genetic variability. So sometimes alleles are just lost from the gene pool completely. Using sort of that other example, I'll just back up. Using this example, this was an example of genetic drift. So put that out of your brain for a second. Imagine we're talking about, we are talking about genetic drift, but again, imagine now we're specifically talking about bottleneck effect. So let's just ignore these other two generations and let's just look at this one generation. And instead of talking about which ones are surviving or mated to produce the next generation, let's just imagine, I don't know, there was some kind of hurricane that came through, and this is a population of flowers that's on an island. And let's just imagine randomly when the hurricane came in, it killed all these flowers that I'm Xing out, right? It's killing all of them. Not because there's anything wrong with those flowers. It just so happened that that's what the hurricane randomly took out. Now we have this new population of the ones I'm circling, right? They're all big R. So in this hypothetical situation that I'm trying to show you, randomly, all the other flowers were killed. So randomly, we're left with big R, right? We lost the little r allele. And that is one of the things that can happen. And of course, that might not seem like a big deal when you're talking about flowers. Like, okay, a big deal. So now we have red flowers instead of white flowers. But later in the semester, we're going to talk a little bit more about something called biodiversity. And part of that includes all the different alleles in the gene pool. So later you're going to learn why genetic variability is so important. So for now, I'll just say, hey, it's a bad thing. It's not good. And later we'll talk more about why it's not good. Also, when a population goes through a bottleneck, it results in a loss of individual variation, meaning, the individuals that are left are going to be very similar to each other, which means they reduce their adaptability. So again, like I said, with this first bullet point, where's my pointer at here? When I was talking about this bullet point, I said, genetic variability going down is a bad thing. Later in the semester, we're going to talk about why, and we are. But this bullet point is basically a preview of why it's a bad thing, right? So again, later in the semester, this second bullet point, we'll really get into it. But for now, again, just the idea of individual variation, it's a good thing, right? Because that way, if you have a lot of variation in your population, then as things change, right, as the environment changes, the world changes, then you've got all these different variations of individuals, some of which are going to be more likely to survive this change, right? And that's why it's an important thing in a nutshell. But again, we'll talk about it more later in the semester. Are there any questions about what's on the slide now before I show you like a visualization of what's on the slide? All right, so this is from the old textbook, and I kept it. I think it's a great visualization. So imagine this is the gene pool of the original population, or if you want to, just think of it as the original population. They're all the same species, but there's orange. That's not orange. Excuse me, maybe gold, green, purple, right? So there's 3 different versions of whatever this population is. Sort of like the goldfish, right? Again, there's orange, there was white, there was spotted. So here we go. This original population has 3 different versions of whatever. Then it goes through a bottleneck effect, right? So you're going to lose most of the population. And randomly, not because purple was had any kind of advantage, but randomly, your new population is almost entirely purple with one green, and you completely lost out on the gold, right? And again, this is the bottleneck effect, but the bottleneck effect is a type of genetic drift. So this was all random. There was no advantage to being purple, no disadvantage to being orange, gold or green. So any questions about this slide? Okay, let's move forward. At the beginning of the beginning of the chapter, I told you we were going to talk about this later in the chapter. So here we are talking about it. So we're still talking about the bottleneck effect, and a real life example of that would be cheetahs. So cheetahs have gone through two bottleneck effects that we know of. One was in the last ice age, which was about 10,000 years ago, and then the more recently there was farmers nearly killed them all in the 1800s. So twice they almost went extinct, and because of that, they have a very shallow gene pool, so to speak, right? They don't have much genetic variation. And again, later in the semester, we're going to talk about this idea of biodiversity and why it's so important. But yeah, so that's the result, right? The bottleneck effect will result in a smaller population that has less genetic variability. So make sure you're just aware of that. That's definitely going to be a test question, some version of that, right? The bottleneck effect will result in a population that has less genetic variability. So even, again, using these cheetahs as an example, even when the numbers rebound, right? So even, if this was a graph, and we have time and years on the x-axis, and the number, right, of cheetahs on the y-axis, even after they go through this bottleneck effect, the population crashes, and then maybe eventually it gets back up to where it was, right? So you might be back at the same number where you started, but you still have that little variability, right? You still have a shallow gene pool. So even though your numbers may recover, it's still not the same population, right? The population has less biodiversity. So anyway, if there's no questions about that, we will move forward. And again, that's just an example. For extra credit, I invite you to look up what are some other real life examples of, genetic drift. I haven't even mentioned that, right? So we gave the purple or the white flower versus the red flower example, but that was hypothetical. So if you want to look up a real life example of genetic drift, look up a real life example of the bottleneck effect, look up a real life example of any of these that we're about to talk about. But I'll remind you, if you do that, make sure you include a link to your source. So anyway, let's talk about the next one, which is founder effect. And I'm hoping this is easy for you because it's in the name. So sometimes in biology, the names actually make sense. And I hope that makes things easier for you guys. But yeah, the founder effect. This is when a few individuals colonize an isolated habitat. So in a sense, when you think about it, very loosely, this is almost like the bottleneck effect, right? Because you've reduced the population the way it's different, and you're going to need to know this for the exam, right? So instead of reducing the original population to a small number, it's more like we're starting a new population with a small number, right? It's not like the original population, most of it died out. In this case, we just have a small number of the original population, and they've gone somewhere new to start a new population. There's some real life examples. If you download this PowerPoint, you can click on those and watch videos about them. Obviously, if you do extra credit, you can't use those examples because those are real ones. But yeah, what I like about these examples is these are human examples. So there's these people. This is an island in the Pacific, and the people who happened to colonize it thousands of years ago, or however long it was, they just happen to have a slightly higher percentage of people who are colorblind, right? And because of that, to this day, the population of people who have color or who are colorblind on that island is higher than the human population. Again, it's completely by chance. Obviously, there was no natural selection there, right? There wasn't like these people who are colorblind had some sort of advantage. They just happened to be the ones that populated the island. The Amish, that's a really great example because, you can see here, these other two examples I'm giving you are islands, right? Martha's Vineyard is an island. Pinglap is an island. The Amish are not on islands as far as a piece of land surrounded by water, but culturally they're on an island in a sense, right? When it comes to the Amish people, there's not a lot of interbreeding, right? So that, that's their little gene pool. There's not a lot of people coming into the Amish community, right, including their genes, right? So because of that, in a sense, the Amish are kind of isolated. Now we'll give you one example for the Amish, right? So the percentage of people who have extra fingers and toes, it's called polydactyly. They have a higher percentage in the Amish community than the rest of the human population, because by chance the people that started the Amish community happen to have a higher percentage. And of course, then you get inbreeding and things like that, but that's a whole different discussion. But anyway, for extra credit, I do invite you to look up some other Amish examples, right? What other things are more prevalent in the Amish community versus the rest of the human population, other than polydactyly? And then finally, Martha's Vineyard, which is where ASL, American Sign Language, was started. But Martha's Vineyard to this day has a slightly higher population of people who are deaf than the rest of the human population, because, again, the people who happened to found, started Martha's Vineyard had a slightly higher percentage of people who are deaf. And there you go. So anyway, that's the founder effect. Again, I invite you to look up some other examples, maybe some non-human examples of founder effect. But yeah, and really quickly, sorry, before I move forward, if you remember the conversation we had when we were talking about the Galapagos Islands, and how they were far enough away from the mainland of South America that, individuals were not regularly leaving South America and going to the Galapagos, right? But it was close enough to where it could happen every once in a while. So the Galapagos Islands are just another great example, a broad example of founder effect, right? So the few individuals that made it to the Galapagos Islands, whatever species you might be talking about, were, that's them. That's that new small gene pool. But anyway, any questions about founder effect? All right. Next word for attendance is going to be chesty, Chesty, like Chesty Puller. It's one of the most famous Marines ever. Let's talk about gene flow. I feel like we can finish this day off with gene flow. Hopefully we'll be able to finish that in time. And then on Wednesday, we'll just come back and talk about natural selection again, but we've already talked about it. So anyway, gene flow is basically just a migration, right? And again, I'm calling it gene flow. We called it gene flow in the lab and on that website that I didn't create, right? It's called gene flow, but for some reason your textbook calls it migration. So make sure you take note here for the exam. Those words need to be interchangeable for you. Migration or gene flow for you needs to be the same thing. So when you get a question about migration, you should think, yeah, this is gene flow, or vice versa, right? If you get a question about gene flow and maybe you study a lot from the book and you need to remember, yeah, that's migration. Anyway, this is another source of evolutionary change, and it's just like it sounds, right? It's the genetic exchange with another population, right? It may result in a gain or loss of alleles, right? So, again, if you just have a lot of fish leaving, we'll use that the lab as an example. If you had fish leaving, which we didn't, remember our parameters were set such that we had fish coming in, right? We had orange fish coming in, but we didn't have any leaving. But so you can imagine if you have individuals leaving and not coming back, you could lose some alleles. Conversely, if you have individuals coming in from a different population, they might be bringing in new alleles, right? So yeah, that's gene flow. It also reduces the genetic differences between populations. So again, let's talk about the Galapagos Islands. Imagine if there was a species of bird on one island, and maybe they have they're just really fluffy, right? Really big fluffy feathers. And then a population of the same species on a different island. And they're really slick and not really puffy feathers, right? But they're birds, so they can make it from island to island, and occasionally they do. And over time, you might get those fluffy birds might become a little bit less fluffy, and maybe those slick birds become a little bit fluffier, right? Over time, those genetic differences would reduce. Anyway, that's gene flow. If you download the PowerPoint, you can watch this little short video about gene flow. Are there any questions about gene flow? Perfect. So we've got what, 3 minutes? So yeah, a little bit less than 3 minutes. So that's it. Again, make sure you send in those words. Don't forget to ask one question about the material. It's important. I know some of you don't do it, so it's not just that you're getting a deduction in points, but also it helps me because sometimes you ask an important question. And I'm like, oh yeah, you need to know that, and I'll make sure you know. Or sometimes you might ask me a question. I'll say, hey, you don't need to know that, which is useful, right? So when you're studying for the exam, you can be like, oh yeah, when I asked him this question, he said I didn't need to know that for the exam. So by 9 a.m., please send me those words. I will be online for office hours if you need me until 10 a.m. And I hope you guys have a great week.
Excuse me.
Do you have a question?
Yes.
All right.
Regarding to next week, lab 13. I already kind of got like a start to it, and I'm not gonna be here that day, so I was wondering if we could meet so I can do it sometime this week, considering I already
Sure.
lab 12.
Yeah, whenever you want. You're talking about the ecological footprint lab, right? You already got started on that?
Yeah.
Okay, good. Yeah. Yeah, we can do that whenever you want.
Okay. Thank
It,
you.
it really is simple, to be honest. It's just between I mean, there's 3 of us here right now, but to be honest with you, one of the things is the TAs are required to teach at least one lab. So that's going to be his lab. It's nice and easy. So all that pre-lab introduction stuff you probably already did, that's the stuff he's going to teach. So, and again, like you said, if somebody turns it in before that date, they don't have to come in. And if you can do this, if you want to, you can join us online instead of in person if you want. So yeah, whatever you want to do, just let me know.
Okay, I was gonna ask if we can do it next Wednesday since I already did. I mean this Wednesday since I already did the lab for this Wednesday.
Yeah, that'd be a great idea. So I will be actually. I don't know where I'll be. Well, wherever I will be on Wednesday, I will definitely be online. So that's a perfect time, right? So during the lab portion, that you and I can meet and do the next week's lab. And anybody else, do you mind if I invite other people?
No, I don't mind at all.
All right, so I'll let other people know if they want to join us for next week's lab this week. Then they can join us online and do it.
Okay, thank you.
My pleasure. Have a good
Yeah.
week.
You as well.
Gross, did you need anything?
No, I was just sitting here. Sorry.
All
All
right.
good. Thank
Well,
you.
if you need
You have-
anything, I'll be on until 10. And don't forget to send those words in.
Yeah, I won't. Thank you. Have a good day.
You're welcome.
Bye.
Have a good week.